This video is about immunology. So, immunity in health and diseases, principles and applications of cross reactivity, precipitation, agglutination, and SPR. And this particular study note is developed by EPG Panchala team, Government of India, MHRD. So, I would like to express my sincere gratitude to the development team for developing this study note. Some important aspects related to antigen antibody interactions, basic characteristics of immune reaction, detection of antigen antibody reaction, that factors that define them. Then principles of agglutination, precipitation, and SPR, avidity, and some other aspects. So let's start. Nature of antigen antibody reactions. Look and key concept. An antibody molecule comprises of Fab portion where the active site consisting the hypervariable regions of the heavy and light chains is located. The antigenic determinant resides in a cleft formed by the active site of the immunoglobulin molecule as indicated by X-ray crystallography studies. Thus, the antigen-antibody interaction can be stimulated by a key, the antigen, which fits into a lock, the antibody. So, we are assuming antigen as a key, and the lock is consisting of antibody. Okay. Non-covalent bonds. The binding of an antibody and antigen is highly specific and involves weak and reversible non-covalent interactions comprising mainly of van der Waals forces, electrostatic forces, H-bond, hydrogen bonding, and hydrophobic forces. Hydrophobic forces. The antigen combines to the antibody at the active site by non-covalent bonds. Multiple bond formation ensures that the antigen will be bound tightly to the antibody. And reversible nature. Antigen-antibody complexes are strengthened by non-covalent bonds, thus making their nature reversible. Affinity and avidity. Affinity, the intensity of the reaction between an antigenic determinant and one active site of the antibody molecule defines the affinity of that antibody. Affinity is the equilibrium constant characteristic of an antigen-antibody interaction. It is the sum of the attractive and repulsive forces effective between the antigenic determinant and the active site of a specific antibody. Most antibodies have extremely high affinity and specificity for their antigens. Avidity. Avidity amounts to the total strength by which an antigen binds to multiple antigenic determinants and multivalent antibodies. Avidity is affected by the valence of the antibody as well as of the antigen and is therefore more than the sum of individual affinities. Hence, affinity is the strength of binding between a single antigenic determinant and its corresponding individual antibody combining site, whereas avidity refers to the overall strength of binding between multivalent antigens and antibodies. Okay. Then specificity and cross-reactivity. Specificity. Avidity is this. Affinity is the strength of binding between a single antigenic determinant and its corresponding individual antibody containing one combining site, whereas avidity refers to the overall strength of binding between multivalent antigens and antibodies. Specificity and cross-reactivity. Specificity. Specificity is defined as the ability of a particular antibody active site to recognize and interact with only a single antigenic determinant, or the ability of a population of antibody molecules to react with only a single antigen. Antigen-antibody reactions possess extremely high degree of specificity. Antibodies are capable of discriminating between the primary structures of an antigen, isomeric forms of an antigen, secondary and tertiary structure of an antigen. So that kind of specificity will be there. Then cross-reactivity. Cross-reactivity is the capability of a particular antibody active site. Okay, cross-reactivity is the capability of a particular antibody active site to react with more than a single antigenic determinant, or that of a population of antibody molecules to react with multiple antigens. Cross reactions are multi-specific interactions arising due to sharing of an epitope by a cross-reacting antigen and the immunizing antigen, or cross reactions are multi-specific interactions arising due to strong sharing of an epitope by a cross-reacting antigen and the immunizing antigen or because the epitope is structurally similar to one of the immunizing antigen. Then cross-reactivity usually occurs among polysaccharide antigens containing similar oligosaccharide residues. For instance, the ABO blood group antigens are glycoproteins expressed on the surface of erythrocytes. Factors distinguishing the blood group antigens A and B include fine variations in the terminal sugar residues of these surface proteins. A person with type A blood has anti-B antibodies, a type B person has anti-A, and a type of O person therefore has both anti-A and anti-B antibodies. An individual lacking one or both of these antigens would generate serum antibodies to the missing antibodies. The serum antibody response is not induced by exposure to erythrocyte antigens, but by cross-reacting microbial antigens present on common intestinal bacteria. These antigens in turn induce the production of antibodies in individuals lacking the similar blood group antigens on their erythrocyte surface. These antibodies would cross-react with the oligosaccharides on foreign erythrocyte, forming the basis for blood typing and accounting for the essential compatible nature of blood types during blood transfusions. Detecting antigen antibody reactions. So till now we learned some important factors related to the antigen antibody reactions. Some important factors in the sense like terms like nature of antigen antibody reactions. Nature of antigen antibody reactions like Locansky concept, non-covalent bonding between antigen and antibody, reversible nature, affinity and avidity, then specificity and cross-reactivity, detecting antigen antibody reactions, factors affecting measurement of antigen antibody reactions. Antigen antibody complexes are formed under specific conditions of temperature and pH. In order to determine whether an antigen antibody reaction has occurred, the AgAb complexes formed have to be detected by direct or indirect means. A number of factors influence the detection of these complexes. Affinity. Higher affinity of the antibody for the antigen ensures a stable reaction between the two, thus facilitating the detection of the complex formed. Then avidity. Interactions between multivalent antigens and multivalent antibodies are very stable, aiding in the detection. Then antigen to antibody ratio. The formation of AgAb complex is related to the avidity. Interaction between multivalent antigens and multivalent antibodies are very stable, aiding in their detection. Antigen to antibody ratio. The formation of AgAb complexes in relation to the concentration of the antigen and antibody. Therefore, its detection is directly dependent on the ratio in which they are present at a particular instance. And physical form of the antigen. The physical form of antigen plays an important role in detection of its interaction with an antibody. Particulate antigens result in agglutination on reaction with an antibody. However, precipitation of the antigen would occur in case it is soluble in nature when large insoluble AgAb complexes are formed. Another factor is agglutination. The word agglutination comes from the Latin agglutinate, meaning to glue, referring to clumping of substances. So agglutination is defined as the visible clumping of a particulate antigen when mixed with antibody specific for it. Agglutination is defined as the visible clumping of a particular antigen when mixed with antibody specific for it in the presence of electrolytes at an appropriate temperature and pH. So antibodies are capable of binding multiple antigen molecules, linking them to create a large lattice-like complex which is visible to the naked eye. Antibodies that generate such reactions are called agglutinins. So all antibodies can theoretically agglutinate particulate antigens, but IgM, due to its high valence, is particularly a good agglutinin. Large antigen with multiple epitopes easily adhere to particles such as animal cells or bacteria when combined with specific antibody, resulting in cross-linking. The process of agglutination involves two steps. First step is sensitization and second is lattice formation. Sensitization is recognition and attachment of specific antibody to corresponding antigen. Temperature, pH, and time of incubation influence the reaction. A lattice is formed by cross-linking between sensitized particles. Agglutination reaction used for diagnosis of disease in lattice lab either uses the particulate or soluble antigens. Example of agglutination reaction using particulate antigen is Salmonella typhi bacteria to detect specific antibody in serum from patients suffering from typhoid fever. Widal test. An agglutination is a serological reaction similar to precipitation with the exception of the antigen being large and particulate in case of agglutination. Both reactions are inhibited by antibody excess and this phenomenon is called the pro-zone effect, whereas in case of antigen, excess post-zone effect occurs. If the antigen is an integral part of the surface of a cell or other insoluble particle, the agglutination reaction is known as direct agglutination. If the antigen is an integral part of the surface of a cell or other soluble particle, it's called direct agglutination. However, a cell or insoluble particle can be coated with a soluble antigen such as viral antigen, a polysaccharide or a hapten, and the coated antigen can be used in an agglutination test for antibody to the soluble antigen in a reaction called passive agglutination. So, two types are there, active agglutination and passive. Pro-zone effect, whereas the case of antigen excess, post-zone effect occurs. If the antigen is an integral part of the surface of a cell or other insoluble particle, the agglutination reaction is known as direct agglutination. However, a cell or insoluble particle can be coated with a soluble antigen such as a viral antigen, a polysaccharide or a hapten, and the coated cells can be used in agglutination reaction is known as direct agglutination. However, a cell or insoluble particle can be coated with a soluble antigen such as a viral antigen, a polysaccharide or a hapten, and the coated cells can be used in an agglutination test for antibody to the soluble antigen in a reaction called passive agglutination. So, Epitopes IgM bacterium. Agglutination. Pentavalent immunoglobulin IgM is shown to interact and bind with multiple antigenic epitopes on the surface of bacterial cells, causing clumping or agglutination reaction. Agglutination. Pentavalent immunoglobulin IgM is shown to interact and bind with multiple antigenic epitopes on the surface of bacterial cells, causing clumping or agglutination reactions. Agglutination inhibition. In agglutination inhibition, absence of agglutination is diagnostic of an antigen. Agglutination inhibition. A modified agglutination reaction is a highly sensitive assay for detecting small quantities of an antigen. Its applications include the simple type of pregnancy test kit where latex particles coated with human chorionic gonadotropin and antibody to HCG. On reaction with urine from a pregnant woman, which would contain HCG antigen, agglutination of the latex particles is inhibited when the anti-HCG antibody is added. This absence of agglutination preliminary confirms pregnancy. Another instance where agglutination inhibition assay can be used is detection whether an individual has consumed certain types of illegal drugs such as cocaine or heroin. A urine or blood sample from the suspect is first incubated with antibodies specific for a particular drug. Then, erythrocytes or other particles coated with the drug are added. If the erythrocytes are not agglutinated by the antibody, the sample contains an antigen that binds to the antibody, indicating that the individual had consumed the illicit drug. However, some legal drugs have chemical structures like those of illicit drugs and may cross-react with the antibody, giving a false positive reaction. Therefore, a positive reaction is a preliminary test followed by a non-immunologic method for confirmation. Qualitative agglutination test. Agglutination tests could be performed in a qualitative manner to assay for the presence of an antigen or an antibody, and the antibody is combined with a particular antigen. The agglutination of this antigen indicates a positive test. For an individual's serothymocytes can be mixed with antibodies specific to different blood group antigens to determine his blood type. Alternately, an individual's serum is mixed with erythrocytes of a known blood type to assay for the presence of antibodies to that blood type in the person's serum. Then quantitative agglutination test. Agglutination tests could also be quantitative, where the level of antibodies to particular antigens can be measured. This test is performed by making serial dilutions of the sample to be tested for antibody, and then a fixed number of erythrocytes or bacteria or other such particulate antigen is added. The maximum dilution that gives visible agglutination is then determined and is known as titer. So, agglutination tests could also be quantitative, where the level of antibodies or particulate antigens can be measured. This test is performed by making serial dilutions of the sample to be tested for antibody, and then a fixed number of erythrocytes or bacteria or other such particulate antigen is added. The maximum dilution that gives visible agglutination is then determined and is known as titer. The results are reported as the reciprocal of the maximum dilution that gives visible agglutination. Hemagglutination. Hemagglutination reactions involve agglutination reactions using erythrocytes. These reactions are used in blood typing, diagnosis of certain diseases, and identification of certain viruses. Blood typing tests with the ABO antigens involve mixing the RBCs on a slide with antisera to the antigens A or B. If the corresponding antigen is present on the cells, agglutination results in the form of visible clumping on the slide. Identification of the antigen present on donor-recipient RBC is the fundamental principle for matching blood types for transfusions. Application of agglutination tests. Determination of blood group types or antibodies to blood group antigens. Assessment of bacterial infections. Infection with a particular bacterium is indicated by the rise in titer of an antibody to this bacterium. In Coombs test. When erythrocytes react with antibodies, agglutination may not always result. It could be due to deviation from antigen-antibody ratio from its optimal concentration, or zeta potential on the erythrocytes could be preventing cross-linking of cells. In order to detect non-agglutinating antibodies on erythrocytes, a second antibody directed against the antibodies attached to their respective epitopes on erythrocytes is added. Antibodies of one species are immunogenic when injected into another species, leading to production of anti-immunoglobulins. Many anti-immunoglobulins bind with antigenic determinants present on the Fc portion of the antibody and leave the Fab portions free to react with antigens. Many anti-immunoglobulins bind with antigenic determinants present on the Fc portion of the antibody and leave the Fab portion free to react with antigens. Then precipitation. The smallest unit of an antigen molecule that can bind with an antibody is known as antigenic determinant or epitope. Antigen molecule that can bind with an antibody is known as antigenic determinant or epitope, which is present on the antigen. Okay, antigen epitope. So antibody paratope. So the corresponding region on the antibody molecule that interact with the epitope is called paratope. Antibody paratope. The number of epitopes on the surface of an antigen is known as its valence, and it determines the number of antibody molecules that can combine with the antigen at the same one time. Monovalent antigens are those having a single epitope. However, more than one copy of the same epitope is present on most antigens called polyvalent antigens. So, immunoprecipitation involves interaction of a soluble antibody with a soluble antigen, resulting in the formation of an insoluble product, the peptide precipitate. These reactions consist of lattice cross-link formation when the corresponding antigen and antibody combine in optimal ratios. Lattice formation relies upon the valency of both antibody and antigen. Cross-linked complexes result when bi or polyvalent antigens interact with more than one multivalent antibodies. If the Ag-Ab complexes formed are too large to stay in solution, visible precipitation results. Excess of either component residues, lattice formation, and subsequent precipitation, hence, this could occur at optimal concentrations. Antibodies that aggregate soluble antigens are called precipitants. So, antibodies that aggregate soluble antigens are called precipitants. Precipitation and agglutination reactions differ in size, solubility of the antigen, and sensitivity. Antigens are soluble molecules and larger in size in precipitation reactions. Antigen-antibody lattice formation is governed by the valence of both the antibody and antigen. The antibody should be polyvalent, Fab fragments in order to form a precipitate. The antigen should be maybe or polyvalent. It should possess at least 2 copies of an epitope or have different epitopes that are capable of reacting with various antibodies in a polyclonal antisera. Antigen antibody, zone of antibody excess, precipitate formed, zone of equivalence, zone of antigen excess, antibody precipitate, antigen added. Interaction of antigen with specific antibodies generate the precipitate. Different zones are highlighted according to the concentration of different distributed components. Precipitation reactions in fluids yield a precipitin curve. When a constant amount of antibody is taken in a series of tubes and increasing amounts of antigen is added to these, variable amounts of precipitates form, resulting in the quantitative reaction. When a constant amount of antibody is taken in series of tubes and increasing amounts of antigen is added to these, variable amount of precipitates form, resulting in a quantitative reaction. Initially, this method was used to determine the amount of antigen or antibody present in a particular sample. After precipitation, the tubes are centrifuged to pellet the precipitate, and the amount of precipitate is measured on removing the supernatant. The amount of precipitate when plotted against increasing antigen concentrations yields a precipitin curve. Excess of either antibody or antigen interfere with maximum precipitation. Hence, formation of an insoluble antigen-antibody complex occurs within a narrow optimal concentration range known as zone of equivalence. From this, we can see zone of equivalence, zone of antibody excess, zone of antigen excess, precipitate formed along with zone of equivalence. This zone represents the conditions under which antigen-antibody complexes formed are sufficiently large to be precipitated. At equivalence, a large multimolecular complex is formed which increases in size and precipitates out of solution. On the other hand, outside this zone, antigen or antibody excess occurs, resulting in the formation of small soluble complexes. Antigens soluble, zone of equivalence, visible precipitate, antibodies, precipitation band. Test tube reaction for antigen-antibody combination, both reactants migrate towards each other and a precipitation band is formed in the zone of equivalence. Immunoprecipitation reactions in gels yield visible precipitin lines. Immunoprecipitation reactions in gels yield visible precipitin lines. Immune precipitates obtained on an agar mix matrix are referred to as immunodiffusion reactions. When antigen and antibody diffuse towards each other on a solid matrix, or when antibody is incorporated into the gel and antigen diffuses into the antibody containing matrix, a visible line of precipitin line would form. Visible precipitin reaction occurs in the zone of equivalence, whereas no reaction is visible in antibody or antigen excess regions. Different types of immunodiffusion reactions are used to determine relative concentrations of antibodies or antigens, to compare antigens, or to estimate the relative purity of an antigen, such as radial immunodiffusion, Mersenne method. Radial immunodiffusion is a simple quantitative technique where a serially diluted antigen present in wells diffuse into the gel matrix containing uniformly distributed antibody. Radial immunodiffusion is a simple quantitative technique whereas serially diluted antigen present in wells diffuse into the gel matrix containing uniformly distributed antibody. When the antigen diffuses into the gel, it reacts with antibody as soon as it encounters the latter. This is followed by attainment of the equivalence point, visualized at the form of ring of precipitation around the well. The diameter of the precipitation ring is directly proportional to the log concentration of antigen since the amount of antibody is constant. Thus, the amount of an antigen in an unknown sample can be quantitated from a standard curve obtained from different concentrations of a standard antigen. If more than one antigen-antibody reaction has occurred, corresponding number of multi rings would form indicating that the sample contains a mixture of antigens or antibodies. The test is commonly used for the determination of immunoglobulin levels in clinical samples. So, antigen diffusion, antibody incorporated in agar, antigen precipitate forms ring. That is radial immunodiffusion. A precipitin ring formed as a result of antigen-antibody interaction is visible in the agar matrix in the form of a radial sphere. Immunoelectrophoresis. Immunoelectrophoresis involves placing a complex mixture of antigens in a well, and electrophoresis on an agar gel so that the immunoelectrophoresis involves placing a complex mixture of antigens in a well, and electrophoresis on an agar gel so that the antigens are separated according to their charge. Following electrophoresis, a trough is cut in the gel and antibodies are added. The antibodies diffuse into the gel, resulting in formation of precipitin lines. So the antibodies diffuse into the gel, resulting in formation of precipitin lines in the equivalent zone as a consequence of the antigen-antibody reaction. This test provides quantitative analysis of complex mixtures of antigens along with a crude measure of quantity represented by the thickness of the line. The components in a patient's serum are commonly analyzed using this test, where the serum is placed in the well while antibody to hold serum in the trough. Countercurrent electrophoresis, double immunodiffusion, Aucherloni. Aucherloni test, named after the Swedish physician who invented is one of the most commonly used serologic precipitation reactions and is based on double immunodiffusion, also known as countercurrent electrophoresis. It involves placing the antigen and antibody in wells punched in a gel matrix and allowing the two radially diffuse into each other, hence producing a concentration gradient. So, different geometrical patterns have been observed in precipitin lines formed between the antigen and antiserum wells. The patterns of these lines are indicative of the amount of similarity or dissimilarity between the antigens. Rates of diffusion of these molecules increase in proportion to their concentration in the wells, but decrease in proportion to their sizes. Only antigen and antibody having opposite charges can be used for this reaction. This test is primarily qualitative, although semi-quantitative analysis can be performed by analyzing the thickness of the line. Its major advantages are its speed. So you could see a picture which depicts the Aucherloni technique. Double immunodiffusion. The antigen and antibody diffuse towards each other in gel matrix and they interact to form a precipitate in the zone of equivalence. Precipitate formed antibody, agar matrix, antibody travel to antigen, antigen travel towards antibody. Then in the equivalence region, precipitate is formed. Immunologic relationship between two antigens can be analyzed using this technique. Identify occurs when two antigens share similar epitopes. Non-identity occurs when two antigens are discrete and unrelated, share no common epitopes. The antisera form an independent precipitin line on each antigen and the two lines cross. Partial identity occurs when two antigens share same epitope, line of identity, but one of them also wears a unique epitope, curved spur. This technique can also be used to estimate the relative concentration of antigens. The distance of formation of the precipitin ring from the antigen well is indicative of concentration of the antigen. Higher the concentration of antigen, greater is the distance between the equivalent zone and the well. Antigen, antibody, identical, non-identical, partially identical. Aucherloni test, the precipitin lines from differentiable patterns in different antigen-antisera interactions. Relationships between the reacting components can be identified on the basis of the nature of precipitin line produced. Then SPR, surface plasmon resonance. SPR is a non-invasive optical technique that involves label-free means of real-time tracking of binding of an injected analyte to an immunized biomolecule. So analyte binding leads to a subsequent increase in mass and thus proportion increase in refractive index observed as a shift in the resonance angle. The SPR procedure involves a flow injection comprising the analyte of interest dissolved in a buffer, transversing the sensing surface to network with the immobilized biomolecule. SPR transducers operate by prism coupling incident light onto an optical substrate coated with a semi-transparent noble metal AU. Total internal reflection TIR occurs at this surface such that all of the light is reflected while none of it refracted above a certain incidence angle. First TIR should occur at the interface until the incident light drops to the critical angle at which a fraction of the light is refracted across the interface. At the point of reflection at the interface, an evanescent field standing wave would penetrate the exit medium to the order of 1/4 of the incident light wavelength. Light source, optical detector, sensor tip, glass rod, ligand, analyte, intensity response, surface plasmon resonance. The basic principle of technique is highlighted. Coupling of the incident light with the surface plasmon affects the intensity of the reflected light and patterns are analyzed. When monochromatic polarized light strikes the interface of two transparent media, in this case glass prism and the buffer solution from the side of media with the highest refractive index, glass prism, the light is partially reflected and partly refracted towards the plane of the interface. The coupling of the incident light to the surface plasmons leads to loss of energy and thus a reduction in the intensity of the reflection light. The technique uses a photo detector array to measure very slight deviations in SPR. The output can be viewed on the BIA core as dips. The deviations in diffractive index are recorded in real time and plotted as resonance units versus time. A background response is usually produced if there is a difference in the refractive indices of the running and sample buffers. The response value must be deducted from the anti-sensogram to obtain the actual binding response. Advantages of SPR: ability to perform real-time measurement, eliminate the need for labeled reactants, exceptional sensitivity, small quantities of purified reagents are required. Disadvantages of SPR: lack of sensitivity when monitoring, low molecular weight absorbate, a rate limiting factor of mass transport affecting kinetic analysis, coupling to AFM, coupling with mass spectrometry. In summary, antigen-antibody interactions depend on 4 types of non-covalent interactions: hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Cross reactions are multi-specific interactions arising due to sharing of an epitope by a cross-reacting antigen and the immunizing antigen, or because the epitope is structurally similar to one on the immunizing antigen. The interaction between a particulate antigen and agglutinating antibody like agglutinin produces visible clumping or agglutination that forms the basis of simple, rapid, and sensitive immunoassays. The interaction of soluble antigen and a precipitating antibody in a liquid or gel medium forms an AG-AB precipitate. Electrophoresis can be combined with precipitation gels in a technique called immunoelectrophoresis. Precipitation reactions are based on the interaction of antibodies and antigens, two soluble reactants that combine, resulting into one insoluble product, the precipitate. These reactions involve formation of lattices, crosslinks. SPR is a non-invasive optical phenomenon that allows label-free means of observing interactions occurring during binding of an injected analyte to an immunobilized biomolecule in real time.